So, welcome everyone, and thanks for joining on time. Those who have joined already, and it's the time was actually 7:30, and it's already 32. So I think we can start. And along with everyone will join. They are all welcome. So today uh, our presenter Arbid Hassan. Hello, hello.
Hello, yeah. Can you hear me
Yeah,
properly?
yes, I can. Thank you. I can see you have started sharing your screen, and I can see your slide properly. So I can think you can start, and looks like a good paper. So I guess everyone will enjoy it pretty much I will be. So you can continue your presentation, and thank you again.
Okay, so hello everyone. I'm Arbid Hassan and I have recently graduated from Northside University and my major was in biochemistry and biotechnology. So today I'm going to present about an exciting topic which is about obesity, that how obesity dysregulates the pulmonary antiviral immune response. This paper was published in Nature Communications. Now moving on to the abstract part. In this study, they explored the connection between obesity and severe influenza infections, focusing on the pulmonary antiviral immune response. They discovered that obese individuals exhibit deficient immune response in bronchoalveolar lavage cells, unlike in bronchial epithelial cells or blood dendritic cells. A significant finding was the disturbed airway metabolism in obesity, in particularly the increased leptin levels, which is inversely correlated with antiviral response. Further on, the experiments they performed in mice confirmed that heightened leptin directly hinders type 1 interferon response, and observation in obese patients hospitalized with influenza further supported these findings, suggested potential therapeutic avenues like leptin manipulation or interferon administration for better protection in high risk of obese populations. Now, let's know about the global situation about obesity. So the global obesity crisis is impacting over 650 million people and has been linked to an increased risk of severe respiratory viral infections. As seen in the H1N1 pandemic, which happened during 2009 and COVID-19. Studies highlight a compromised immune response in obese individuals, notably in the airway macrophages. There is a deficiency in interferon responses. And this immune impairment is partly due to altered airway metabolomics, especially increased leptin levels, which affect macrophage function. And these findings underline the critical role of macrophage-mediated immunity in the heightened vulnerability of obese individuals to respiratory viruses. So what is the objective of this study? So the objective of this study is to investigate the mechanism by which obesity contributes to an increased risk of severe respiratory viral infections, particularly focusing on the innate antiviral immune response in obese individuals. And this study also aims to understand that how obesity impacts the production of type 1 and type 3 interferon and their role in pulmonary mucosal protection against viruses like influenza, with a specific focus on different immune cell types, such as macrophage, epithelial cells, and dendritic cells. So this now we'll see about the methodology and the study design and participants. So the ethics committee approvals obtained from relevant committees for human and animal studies, since this study included animal research as well. So this study included morbidly obese and healthy non-obese individuals matched by age, gender, and ethnicity. There were around 15 group of individuals. Subjects underwent comprehensive clinical sampling, including blood, nasal, and bronchial samples. Various cell types were isolated for the study, such as the bronchoalveolar lavage cells, bronchoalveolar lavage macrophages, bronchial epithelial cells, and dendritic cells. So the experiments involved ex vivo virus infection and in vitro analysis of cell responses. And animal experiments included leptin treatment and influenza infection in mice. Besides that, real-world patient data from the mechanism of severe acute influenza consortium study of hospitalized influenza cases were also analyzed. An extensive metabolomic analysis of the bronchoalveolar lavage samples were conducted. Besides that, various statistical methods were also applied for the data analysis, including Mann-Whitney, U-test, and ANOVA. So now let's move on to the results section. So their primary finding was that obesity does not alter the bronchial epithelial immune responses. So the study investigated the impact of obesity on the bronchial epithelial immune responses to influenza, despite the known impairment of type 1 and type 3 interferon in respiratory diseases like asthma and COPD. This research found no significant difference in interferon response in bronchial epithelial cells between obese and non-obese individuals. So this says that various influenza strains, besides that, they use various influenza strains to stimulate BECs, and the results showed no notable induction of interferon alpha. However, interferon beta and interferon gamma 1 were induced by the H3N2 strain in both groups, which is basically the strain of the influenza. And this indicates that the obesity does not alter epithelial, antiviral, or pro-inflammatory cytokine response to influenza. Now, their second finding was like antiviral immune responses are impaired in bronchoalveolar lavage cells from obese individuals. So the bronchoalveolar lavage cells were from both groups were infected with different influenza strains. Significant reduction in interferon alpha was noted in individuals compared to non-obese persons. And interferon beta and gamma responses were also reduced in the obese subjects. Despite this, no difference was observed in the influenza M gene copies between obese and non-obese individuals. However, obese individuals exhibited lower baseline and post-infection production of pro-inflammatory cytokines like interleukin-6, CXCL8, and interleukin-8 tumor necrosis factor in the bronchoalveolar lavage cells. Moving on, they also checked the role of dendritic cell in the obese and non-obese individual. The study evaluated the dendritic cells from obese and non-obese individuals for antiviral responses. The plasmacytoid dendritic cells, which are known for potent type 1 and type 1 interferon production, were isolated and tested with 3 influenza virus strains. Both obese and non-obese subjects showed significant induction of interferon alpha, beta, and gamma 1, particularly with the H1N1 strain and the B Florida strains. So the magnitude of the interferon production in response to influenza was comparable between the obese and the non-obese groups, which showed that the pro-inflammatory cytokine responses in the dendritic cells were also similar in the both groups. The study concluded that the obesity does not impact the dendritic cells' ability to mount an antiviral immune response. Moving on to the next slide. So this is about like another finding, that alterations of the airway metabolomic and adipokine milieu in obesity. So the study found specific impairment in antiviral immunity within the BL cells macrophage in obese individuals. Analysis of the bronchoalveolar lavage fluid metabolites revealed that altered metabolic profiles in the airways of the obese individual were found, and significant changes in 17 metabolites were noted, with most being downregulated in obese individuals, except for the AMP and glycerol. So AMP stands for adenosine monophosphate, and enrichment analysis indicated altered selenoamino acid, methionine fatty acid metabolic pathways in the obese subjects, and the leptin concentrations were higher in both upper and lower airways of the obese individual. So elevated level of leptins correlated with weaker induction of interferon beta responses in the bronchoalveolar lavage cells, and higher bronchoalveolar lavage AMP levels suggested a link between leptin and impaired antiviral immunity in obesity. They further explored the leptin's role in impaired interferon responses in the mice study, and the mice were administered with recombinant leptin. Leptin alone can increase the lung mRNA expression of the interferon beta and antiviral genes. However, leptin administration before influenza infection attenuated the expression of these antiviral genes. Post-infection leptin-treated mice showed increased pro-inflammatory response and cytokines. And leptin also increased the the SOX3 mRNA expression, suggesting a link between high leptin levels and reduced type 1 interferon response due to the SOX3. a negative regulator of type 1 interferon signaling. So they have concluded that leptin treatment impairs the ex vivo type 1 interferon response in mouse macrophage. So the researchers further explored how leptin affects the macrophage function in response to influenza. They isolated the bronchoalveolar lavage macrophages from mice and conducted an ex vivo influenza infection experiment with varying concentration of leptin. So this result showed that higher dose of leptin significantly reduced the macrophage ability to produce interferon beta and the PKR, which is a key antiviral gene upon influenza infection. Additionally, leptin treatment elevated the SOX mRNA level in macrophage, aligning with the in vivo findings, supporting the concept that increased leptin in obesity negatively impacts the macrophage antiviral immunity as well. Finally, the last part of their finding was dysregulated airway in the obese individual hospitalized with influenza. So the mechanism, the MOSAIC study. So MOSAIC stands for Mechanism of Severe Acute Influenza Consortium. They analyzed the hospitalized influenza patients during intense influenza activity in UK, which was conducted in the hospital situated in Liverpool. Out of 133 patients, 27 were classified as obese, which was around like 20.3% of the population, and no differences were found in the nasopharyngeal viral loads between obese and non-obese patients. Obese patients exhibited increased nasopharyngeal concentration of interferon beta and certain pro-inflammatory cytokines. The serum cytokine levels did not differ significantly between obese and non-obese patients. So this indicates that local immune dysregulation in the upper airway mucosa of obese patients during active influenza infection without systemic circulation involvement. Finally, the public health implication of this study was that the role of obesity in severe influenza infections and the importance of targeted public health strategies, and recognizing the obesity as a risk factor for severe respiratory infections can guide policy and healthcare resource allocation. Besides that, awareness campaigns focusing on the risk associated with obesity and viral infections. The potential therapeutic strategies. So they suggested that exploring leptin manipulation as a potential therapeutic avenue for managing obesity, investigating the interferon administration to enhance immune response in obese individuals, and developing treatment that address the unique metabolic and immunological profile of obese individuals. The challenges and limitations of this study. So difficulties were found in obtaining accurate lower airway samples from obese individuals. There were limitations in translating the findings from animal models to human clinical scenarios. And besides that, the need for larger, more diverse human studies to validate finding is always a prime criteria. The future research of this will include examining the impact of sustained weight loss on restoring antiviral immunity, expanding the scope of research to include the other respiratory viruses and varying degrees of obesity, and also investigate the long-term effects of obesity on respiratory health and immune function. To conclude that, this study highlights a critical link between obesity and the susceptibility to influenza. The insights provided from this research can drive novel therapeutic approaches and public health initiatives. And this also urges a broader focus on obesity treatment and management to mitigate risk of severe viral infection. That's all. Thank you for your attention. Hello. Hello. Am I audible? Okay, so I have completed my presentation. So is there any questions?
Hello, Arbid.
Hello.
Sorry, I wasn't there when you finished your presentation. So,
It's
is
all
it
right.
over?
Yeah, it's
Okay.
over.
Oh, I thought since it was a research paper, it could long more than that.
Okay, so
So,
actually I just picked the results sections rather than the procedures that they included. That's why I shortened it up to provide the
okay,
key informations to the listeners.
got it. But it's better if you give all the deeds, and it's
Mm.
important because methodology and also the procedure is also important.
Well, the methodology part was discussed, that how the studies were performed.
Okay. Sorry, I missed it. I'm actually really sorry. So, we have Al Mamun Shohag and Tanvir. Others couldn't join. They might be outside. So, Shohag, what do you think about the presentation or the paper? Hello? If you can't hear me, you can text me or say something. Okay. Tanvir?
No, I have
Yes.
no question.
Okay. What do you think of his presentation and the paper he presented? আপনার কাছে এই রিসার্চ পেপারটা কেমন লাগলো? আপনি সেটাও শেয়ার করতে পারেন বা তার প্রেজেন্টেশন কেমন লাগলো সেটাও বলতে পারেন। Okay. If no one have any question, I think we
Hello.
hello. Yes, I was saying that I will definitely hear your presentation, and since no one is have any questions, so we can end this. Do you have anything to say, Tanvir and Alamin? Almamun? Okay. So thank you, Arvid, again, and also others who joined for today's Journal Club. Thank you and good night, everyone.
হ্যালো আপু, হ্যালো।
Yes, হ্যাঁ বলুন।
আচ্ছা, আমি এই পর্যন্ত কিন্তু আমাকে কোনো অ্যাসাইনমেন্ট কিন্তু দেওয়া হয় নাই আর কি। তো গ্রুপে পোস্ট করলেন যে অ্যাসাইনমেন্ট।
জি, আপনার নামটা একটু বলবেন? তানভীর।
তানভীর। Yes.
আপনি কি রিসেন্টলি অ্যাড হয়েছেন?
হ্যাঁ, আমি রিসেন্টলি অ্যাড হয়েছি আর কি।
অ্যাকচুয়ালি অ্যাসাইনমেন্ট তো হচ্ছে আপনি 9th ব্যাচে না 8th ব্যাচে? হ্যালো, আপনি কোন ব্যাচে? আচ্ছা, অ্যাকচুয়ালি
সেভেন,
আমি
ব্যাচ 7।
7th। ওকে। আমি আপনাকে কালকে জেনে ডিটেল জেনে আপনাকে